Good morning, my name is Gail Ramírez, and today I'm going to talk about the pathophysiology of COVID-19. First of all, what is COVID-19? It is part of a large family of viruses known as coronavirus that can cause respiratory infections in humans. They are known for being able to evolve within the animals and then be transmitted to humans. COVID-19 refers to the infectious disease itself, caused by the most recently discovered coronavirus. CO stands for corona, vi for virus, D for disease, and 19 for the 2019 novel coronavirus. Novel means that it is a new coronavirus that has not been previously identified in humans. This means that it is different from previously found diseases like the SARS or the MERS. The virus' actual name is SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome Coronavirus 2. It was named like this because its structure resembles the one of the SARS virus. The source of the virus is believed to be the Hunan seafood wholesale market in Wuhan, China. This means that the virus might have a zoonotic origin, meaning that pathogens can jump from animals to humans. It has been related to the bat and pangolin coronaviruses because the genetic sequence of the new virus is 96% identical to the one in bats. However, the type of animal the virus originated from is still not clear. The wet markets have a high risk of viruses jumping from animals to humans because the hygiene standards are difficult to maintain as live animals are kept and slaughtered right there, allowing diseases to spread from species to species really easily. The first case can be tracked to a 50-year-old in Hubei on November 17, 2019, but on March 11 of 2020, the World Health Organization recognized the spread of COVID-19 as a pandemic, meaning that it is affecting the entire world. Not to be confused with the term epidemic, which is the outbreak of a disease that spreads quickly and affects many individuals at the same time. Etiology refers to the cause of a disease. COVID-19 is caused by a coronavirus called SARS-CoV-2. It has a zoonotic origin as coronaviruses are common in different species, the most commonly associated one being the bat species. However, the exact source of the virus is still unknown. Anyone can get the virus, but the following conditions increase the chances of developing the disease. These conditions are known as risk factors, and they include a weakened immune system, high blood pressure, heart disease, lung disease, asthma, kidney disease, obesity, diabetes, cancer, liver disease, smoking, and age, as people over 65 are more likely to get the illness. This means that as the virus is already attacking the immune system, having a weakened one for any of these factors is going to cause more severe symptoms in a patient. Clinical manifestations. The most common symptoms include fever, dry cough, and tiredness. The serious symptoms include difficulty breathing or shortness of breath, chest pain or pressure, loss of speech or movement, and the less common symptoms include aches and pains, sore throat, diarrhea, headache, loss of taste or smell, rash skin, and the discoloration of fingers or toes. Here we can see that the virus is affecting the whole body, for example, causing the nervous system to be damaged, and also the hypothalamus, that way causing fever. Also, the difficulty breathing or shortness of breath is caused by the damage done to the alveoli, and symptoms like diarrhea are caused depending on where the virus binds to which receptors, either on the lungs or the intestines. On average, it takes 5 to 6 days from when someone is infected with the virus to show symptoms. However, it can take up to 14 days. Patients with COVID-19 are classified according to their symptoms. The asymptomatic ones test positive in a test, but they don't have any clinical symptoms and signs of the chest imaging is normal. The mild ones have symptoms of acute upper respiratory tract infections such as fever, fatigue, cough, a sore throat, runny nose, and sneezing, or digestive symptoms such as nausea, vomiting, abdominal pain, or diarrhea. The moderate ones include pneumonia with no obvious hypoxemia. Hypoxemia meaning a low concentration of oxygen in the blood. Severe symptoms include pneumonia with hypoxemia, and the really critical ones include acute respiratory distress syndrome, and they might have shock, myocardial injury, heart failure, coagulation dysfunction, and acute kidney injury. Transmission. The virus that causes COVID-19 is thought to spread mainly from person to person through respiratory droplets produced when an infected person coughs or sneezes, which can travel 6 feet away, which is equivalent to 1.8 meters, and land in the mouth, nose, or eyes of people who are nearby and then inhaled into the lungs. Some people who have the virus don't have symptoms, but they can still spread the virus, which is the reason why this virus has spread exponentially among the population. You can also get the virus from touching a surface or object the virus is on as it remains alive for a relatively long period of time, and then touching your nose, eyes, or mouth. For example, in copper, the virus can stay alive for 4 hours, in cardboard or up to 24 hours, on plastic or stainless steel from 2 to 3 days. COVID-19 is community spread, meaning that people have been infected with the virus in an area, included some who are not sure how or where they became infected. On the first image, we can see how the transmission of COVID-19 works. On the left, we have an infected individual, and when he or she sneezes, droplets are liberated which can be inhaled by another individual, but also droplet nuclei can be formed and someone can inhale that air. There can also be direct contact with the infected person or indirect contact by touching an infected surface. So, the ways in which COVID-19 is transmitted can be for touching your face, by sneezing, shaking hands, or touching infected objects. This leads us to the importance of soap and hand sanitizer. Soap is the most effective way to get rid of the virus. The virus is surrounded by a phospholipid layer which has a hydrophilic head and a hydrophobic tail. Soap molecules have a similar structure, one hydrophilic and one hydrophobic part. The soap molecules get into the membrane and destroy the structure. On the other hand sanitizer contains ethyl alcohol, isopropyl alcohol, or both of them to kill bacteria and viruses on the hands. Alcohols have long been known to kill germs by denaturing their protective outer proteins or microbes by dissolving their membranes. It is recommended that the hand sanitizer has at least 60% alcohol. Some of the main tests that can be done for COVID-19 include the viral test, which can tell you if you have a current infection. They are the most common method, and they look for signals of the virus in the upper respiratory tract. Then the samples are sent to the lab for analysis, and then the results can be obtained. If it tests positive, you can have the disease at the time the test was done, and if it tests negative, you were not infected at the time the sample was collected, but you can still get sick. Then an antibody test can be performed to check the blood by looking for antibodies which can show if you had a past infection with the virus that causes COVID-19. If it tests negative, you might still be infected, as it takes 1 to 3 weeks after the infection to create an antibody. And if it tests positive, you may have antibodies, but it is still not known if people could get infected again or how much time the protection would last. Getting into a deeper level, how do viral tests work? A sample is taken from the inside of the nose or the bottom of the patient's throat, and the sample is taken to the laboratory for analysis. RNA is extracted from the virus and purified, and an enzyme called reverse transcriptase converts RNA into DNA. The DNA obtained is mixed with primers, DNA fragments designed to bind with characteristic areas of the virus genome. Then, by repeatedly heating and cooling a virus DNA mix, primers, and an enzyme that synthesizes DNA, millions of copies of the viral DNA are produced. The more copies of the virus DNA that are produced, the higher the fluorescence is. If the fluorescence exceeds a certain threshold, the test is positive. If there are no viruses, there are no copies of the viral DNA, and therefore the threshold is not passed. Then the test is negative. Some of the problems with this test include a reagent shortage, as the high demand of some reagent problems can delay testing in some countries. Also, tests are slow, as the results can take several hours to arrive, limiting the number of tests that are performed, and also there can be false positives and false negatives. In some cases, deterioration of contamination of the sample can alter the results. Then, some future tests can be performed, like the antibody test. Now, let's talk about the structure of the SARS-CoV-2. Protein spikes on the surface appearing like a crown give the name to the virus. In fact, all of the coronaviruses have this structure present. There are 4 structural proteins in it, which are the spike proteins, the envelope proteins, the membrane proteins, and the nucleocapsid. The spike proteins allow the virus to attach to the membrane of a host cell. They contain a receptor binding domain in order to recognize specific receptors in the host cell, which are the ACE2 angiostensin converting enzyme receptor 2, which is found in the lungs, heart, kidneys, and intestines. The receptor of SARS-CoV-2 binds with an affinity 20 times greater than the SARS virus, which is why it has been really easy to propagate. It also has two function subunits, which are the S1, which is used to bind to the cell's host receptor, and the S2, which mediates the fusion of the viral and cellular membranes. The membrane protein is the most abundant on the viral surface. It defines the shape of the viral envelope, and it is the central organizer for the assembly of coronavirus. The envelope protein is the smallest one of the structural proteins. It is fundamental for the assembly and release of the virus from host cells, and during replication it is largely localized at the site of the intracellular trafficking, especially at the endoplasmic reticulum and the Golgi apparatus of the host cells. Finally, the nucleocapsid protein is basically an RNA binding protein. It helps with the formation of the capsid, it interacts with the membrane protein during assembly, it packages the viral genomic RNA to form the helical nucleocapsid that is incorporated into the budding particle and complete the infection. It is also associated with the replication transcription complexes. Then we have the viral envelope. It is found underneath the surface proteins, it is the virus outer layer, and it is a fatty layer, which means that when it is in contact with soap, it will be destroyed, therefore killing the virus. Then, the capsid, which is a protein shell that encloses the genetic material of the virus. Finally, the nuclear capsid, also known as the N protein, is bound to the virus's single strand of RNA, which contains the genetic information of the virus and allows it to replicate. It also inhibits the defense mechanism of the host cell, and it helps the viral RNA to replicate itself. Here we can see a simpler diagram of the virus and all of its parts. So, can the virus be considered alive? According to the characteristics of life, living things have different levels of organization. Viruses have genes made up of nucleic acids and a capsid made from smaller subunits known as capsomers. Also, living things adapt to their environments. Viruses adapt to the surroundings, going through the lytic phase where the virus actively replicates, or the lysogenic phase, where the viral DNA incorporates into the cell's DNA and multiplies when the cell multiplies. However, there are other characteristics that have to be taken into consideration, for example, homeostasis. Viruses are not made up of cells. A single viral particle is known as a virus. They don't have any nuclei, organelles, or cytoplasm. Therefore, there is no way for them to monitor the internal environment and maintain homeostasis, meaning balance. Also, living things need to reproduce. Since viruses don't have organelles, nuclei, or even ribosomes, they don't have the tools they need to copy their genes, much less create whole new virons. So, they need host cells to reproduce. Living things grow. When a viron is created, it is in its fully formatted state and will neither increase in size or complexity throughout its existence. Living things use energy, and the energy they use comes from the host cells. Also, living things respond to stimuli, and they don't change behaviors in response to touch or sound or light. However, there is still missing research about this topic, so it is not considered a living thing. Pathophysiology of COVID-19. It initially gets into the upper airway through the nasal or the oral cavities, causing the symptoms of headache, stuffy nose, sore throat, and fever. Then, in the mucosal epithelium of the upper breathing tract, the primary replication of the virus occurs. Then the virus goes further down the lungs through the lung epithelial cells, where further viral replication will occur. Once the virus is inside the human body, the life cycle of the virus will consist of 5 main steps. First, attachment. The virus binds to the host cell's receptor. In this step, SARS-CoV-2 binds to the ACE2 receptor through the spike proteins. Most of these receptors are found in the lungs and in the intestines, so depending on where the virus binds, the symptoms that will be presented may be different. If it binds to the lungs, cough is produced, and if it binds to the intestines, diarrhea is produced. Then, penetration. It enters the host cells through the endocytosis or membrane fusion. This happens as the host cells have proteases, which are enzymes that break down proteins. The enzyme divides the spike protein, activating the process of membrane fusion. If the virus is surrounded by the cell, a vesicle is created, allowing it to enter the cell. Biosynthesis. Once inside the host cell, viral contents are released inside the host cell. Viral RNA enters the nucleus for replication, and viral mRNA is used to make viral proteins. This is done using the host cellular machinery, mainly the endoplasmic reticulum and the Golgi apparatus. Then the virus will reach the maturation stage, where specific processes form the envelope glycoproteins, and also a new viral particle is formed. Then we have the release stage, where the new particles are completely made and they are released via exocytosis. Here is a quick summary of the process previously mentioned. First, the virion attaches to the receptors in the cell, and then it is introduced via endocytosis. Then there is an RNA replication, and with the help of the endoplasmic reticulum and the Golgi apparatus, new particles can be made and then be excreted via exocytosis. Then the body will enter a process known as apoptosis, which means cell death. This is caused by the stress the cells are receiving and also by the immune-antigen interaction. Talking about the immune system, in a normal immune response, the immune system kicks in recruiting the white blood cells to clear the virus. In COVID-19, as the virus starts replicating, the immune system tries to stop this, responding with specific antibodies. However, the fast rate at which this occurs ends up harming the organism. The immune system, instead of only getting rid of the virus, enters an overdrive, secreting cytokines, which are inflammatory mediators, leading to a buildup of mucus and thickening and hyperplasia of the cells within the airways, causing irritation of the cells, their lining, and leading to the cough symptoms. This stage is known as the cytokine storm. More about the cytokines. They are proteins involved in cell signaling that mediate the immune responses. There is an exaggerated immune response with a huge release of inflammatory mediators, then causing a cytokine release syndrome. The cytokines are produced by a macrophage inside the alveoli and include the interleukin-1 and the interleukin-6. The secretion of these leads to an increased capillary permeability, resulting in way more fluid entering the alveoli. Also, the plasma accumulates outside the alveoli, compressing the same structure. Increased alveolar edema drowns out the surfactant, which is a substance that reduces the surface tension of a liquid in which it is dissolved. Once there is no surfactant, the surface tension goes up, then collapsing pressure also goes up and the alveoli collapses as well. The uncontrolled response can be the cause of ARDS, which is a rapid inflammation of the lungs. The alveoli walls can become inflamed and thicken, filling the alveoli with fluid, limiting its ability to perform gas exchange within oxygen and carbon dioxide. This is the cause for the shortness of breath and pneumonia symptoms. The inflammatory mediators also bring with them neutrophils, a type of white blood cell that helps heal damaged tissue and resolve infections. When they try to destroy the virus, they release reactive oxygen, that way destroying some of the virus but at the same time the cells inside the alveoli. So, if type 1 cells are destroyed, there is no proper gas exchange, and if type 2 cells are destroyed, there is a decreased surfactant concentration. This forms a consolidation of fluid, proteins, cellular debris, macrophage, neutrophils inside the alveoli, that way also altering gas exchange. So, how are the other symptoms caused? Fever is caused as interleukin-1 and interleukin-6 can travel to the nervous system via the bloodstream. Then, the hypothalamus, which controls temperature, releases prostaglandins, which help regulate the temperature, increasing it and causing fever. Then, cough is caused when the accumulation of things inside the alveoli starts to degrade the body and will try to cough out all of that mucus. Additionally, the immune system can get the disease under control if the virus replicates so fast. Then, a patient would enter a hyperinflammatory phase where the immune system begins to attack itself. The inflammation caused starts limiting the other organs like the heart or kidneys as they stop getting the oxygen needed for proper working. So the overdrive of the immune system is what leads to a severe case of COVID-19. Comorbidity. When it is a very severe case, the inflammatory response can travel to different parts of the body through the bloodstream, leading to a systemic inflammatory response. Then, some other diseases can be caused, like the acute respiratory distress disorder. This is caused by damage to the tiny blood vessels in the lungs, fluid from the vessels leaking to the air sacs of the lungs. These air sacs are where oxygen enters and carbon dioxide is removed from the blood, so when these air sacs fill with fluid, less oxygen gets to the blood. Another disease that can be found is sepsis. This occurs when inflammatory reactions spread through the circulatory system. It causes inflammation in the body, can affect organ functions as tiny clots form, blocking oxygen and nutrients from getting into the blood, and blood pressure can drop, leading to heart failure, stroke, and death. Then there is kidney failure. As the kidneys are not getting enough blood, their ability to get rid of the blood urea and nitrogen decreases, and it can come with cramps, nausea, vomiting, too much urine, or not enough of it. Then there is also liver failure. The liver is not getting enough blood either, and it starts releasing inflammatory enzymes. It can also come with nausea, loss of appetite, fatigue, and diarrhea. Chest CT manifestations of COVID-19. A CT scan or computed tomography can be used as the viral tests for diagnosis can provide false positives. So, how does it work? First, a CT scanner emits a series of narrow beams through the human body as it moves through an arc. Then, the CT scanner's X-ray detector can see hundreds of different levels of density. This data is transmitted to a computer and creates a picture. Some of the pros of it are that CT imaging has a sensitivity of 98%. It is also of great significance while diagnosing the disease, but also when evaluating its progression. However, some of the cons are that they are not specific, meaning that there could be other infections that are causing the disturbances observed in the images. When performing a CT scan, we can see ground glass opacities. There are vague areas with slightly increased density in lungs with obscuration of bronchial and vascular margins, which may be caused by a partial displacement of air due to the partial filling of air spaces or interstitial thickening. Presenting ground glass opacities is the most common imaging finding with occurrence rate up to 98%. Crazy paving pattern can also be observed, which is a scattered or diffuse ground glass attenuation with intralobal septal thickening and intralobal lines. It may be a sign for alveolar edema and inflammatory of acute lung injury. There is a 5 to 36% occurrence in COVID-19 patients, and it may represent a patient reaching a peak stage. Finally, consolidation is another thing that can be observed. This is alveolar air being replaced by pathological fluids, cells, or tissues manifested by an increase in pulmonary density that obscures the margins of vessels and airway walls. There is an occurrence rate of 2 to 64%, and it is considered as an indication of disease progression. It is also a sign for pneumonia. In this slide, we can see a healthy lung CT scan, and as you can see, there is no apparent damage to the lungs compared to the images shown before. Treatments. In severe cases, hospitalization may be needed, where you can receive the following treatments: ventilators, where the patient is connected to a machine facilitating breathing for them, providing them oxygen through small tubes that go into their nostrils; intravenous, where the patient will receive fluids through the tubes that go into his or her veins in order for them not to get dehydrated or malnourished. Medications will be given to the patient to prevent clots. Additionally, they may receive antiviral medicines like remdesivir, which was used towards Ebola, and tocilizumab, used for cytokine release syndrome. Another treatment can be blood plasma. Convalescent plasma therapy consists of the use of blood plasma from people who've recovered from COVID-19 in order to help patients with severe or life-threatening cases, as the blood has antibodies from the virus. No drug has been proved to be safe and effective for treating COVID-19. Trials. There's early data showing that remdesivir, an antiviral drug previously used against Ebola, may be helpful. Other medications that have been used to try to treat severe COVID-19 cases include a combination of lopinavir and ritonavir, both of which are antiretroviral drugs that target viruses composed of RNA rather than DNA, as well as chloroquine and hydroxychloroquine, which are used to treat malaria. There's one trial showing effectiveness of hydroxychloroquine and azithromycin being used together. On March 22, the World Health Organization launched a global mega-trial to test some of these promising medications. How would a vaccine work? A potential vaccine would include faint versions of the virus that would expose the human body to a version of the virus that is weak to provoke an infection but strong enough to stimulate an immune response. Then, the body would start creating specific antibodies for the coronavirus and the spike proteins. These antibodies would attach to the virus and prevent it from binding to the host cells. The immune system would destroy the virus via endocytosis, and all of this is still in research. Talking a little about epidemiology and flattening of the curve, the red curve here represents the COVID-19 cases, and the green line represents the healthcare capacity, which is equipment and personnel. What needs to be done is to flatten the curve, which is done by either increasing the healthcare capacity or slowing the rate at which people is getting sick, like China did. And this is why social distancing is so important. Social distancing is important because if some people continue interacting, there will be an exponential growth of cases, surpassing the healthcare capacity. On the other hand, if lockdown is enforced, the virus would spread to a household level, but then the transmission would end, without the need of overwhelming the healthcare capacity. The origin and evolution of SARS-CoV-2 remains largely unclear. However, some studies have found that there is a sharing of 88% nucleotide identity between the SARS-CoV and the SARS-CoV-2, so they have been taxonomically classified into a single viral species, which is the severe acute respiratory syndrome-related coronavirus. SARS-CoV-2 is closely related to multiple coronaviruses of bats, for example, a bat cov or a bat cov RATG13 detected in the species from China. Two distinct clusters of SARS-CoV-2 related viruses were identified in pangolin samples obtained by the anti-smuggling operations in the Guangxi and Guangdong provinces of China. The Guangdong pangolin coves have a higher amino acid identity of 97.4% with the SARS-CoV-2 than does the bat of RATG13 of 89.2%. RATG13 is more closely related to SARS-CoV-2 than the one in bats of CoV RATG13 of 89.2%. There is also a possibility that the Guangdong pangolin coves come from the cross-species transmission from bats.